and welcome to Brewing Brilliance, a podcast where we talk to academics from across the university who are involved with the Healthy Ageing and Chronic Conditions Research Institute. We will be speaking to researchers who are leading innovative, interdisciplinary approaches to promote lifelong health, delivering improved interventions to manage chronic conditions more efficiently. These sessions are essentially opportunities for us within the Healthy Ageing and Chronic Conditions Research Institute to meet colleagues from across and beyond our faculty to foster knowledge sharing, collaboration and networking. My name is Adrienne Cleverly, and today we are joined by Thomas Wilkinson, who is a Professor of Barrier Immunity and Infectious Disease and lead of the Microbiology and Infectious Disease Group in the Faculty of Medicine, Health and Life Science. Tom joined Swansea University in 2008 and has researched the immune response to infectious disease in humans and animals with the goal of developing early predictors of invasive infection. So hello and thank you for joining us.
Hello. Nice to be here. Hopefully it'll be fun.
Yeah, I think it will. So nice easy question to get you started and ease you into it. Can you tell us a little bit about you and your background?
Yeah, so I'm Tom Wilkinson. I've been at the university for 2008 I arrived as a lecturer and I've been here ever since, and I'm a prof in 2023. But before that I had sort of periods of postdocing in Edinburgh and in Seattle, and I did my PhD in Cardiff. Now most of my work throughout that 25 years then has been involved in studying bacteria and the way bacteria interact with the host. And I sort of been maybe a little bit promiscuous. I haven't stayed in the same area of the body. I've looked at the lung infection, I've looked at bladder infections, gut infections, but essentially it's been a focus on looking at the local infection within different body compartments, and I've done that pretty much throughout my career.
So the infections then are a big part about what you do, and your most recent research is in sepsis, I believe.
Yeah.
So can you just give us a little bit about sepsis? What is it about sepsis that drew you to it?
Yeah, so I talked a little bit about the body compartment. So it's fine to ask about infections in body compartments, but one of the things that you start asking the question is, well, what happens when the infection becomes too much? What happens if there's too many bugs in there? What happens then? And one of the things about body compartments is that they have a nice barrier, an epithelial cell barrier. What happens when that barrier breaks? Well, when that barrier breaks, you get invasive diseases, and a good example of that is bacteremia and sepsis. So sepsis in itself is a very complicated disease, but it's really your body's over-exuberant response to infection. And it involves multiple pathways, inflammatory pathways, complement and coagulation pathways, but it results in multi-organ failure. And this is really a big problem because it doesn't just affect the heart, it affects the kidney, it affects the lungs, it affects all those different parts by your blood coagulating in those different body compartments, which means they don't work anymore.
That's quite good though, because you've got that extensive background. You said you haven't sort of focused on an area, you've done all these different bits. Has that been a benefit to you with working on sepsis, do you think? Or
I think completely it's been a benefit. So there's certainly things that they've got in common. So all those body compartments have got their own way of working, but they all share the idea of the epithelial cell and of the barrier of the epithelial cell, and understanding how you break those barriers, or how bugs can actually break those barriers, or maybe that patients have particular weaknesses that means that their barrier is not so strong.
Yeah,
that's a really great area for research, and that's sort of something that we've touched upon. It's been a recurring theme and has popped back continuously, I think, in my research career.
So is that what sort of encouraged you to sort of look into this? Do you want to tell us a bit about the current research that you've just done?
So the current research, we've got to go back to 2018, I think. So I was having a conversation with one of my colleagues in Hywel Health Board. So that's in West Wales. And so he's in the path lab there, the micro lab. And he had this issue where he saw lots and lots, a very high incidence of E. coli bacteremias. So that's E. coli bacteria, but in the blood. And it was the highest in Wales. One of the reasons for that is probably had a really good surveillance program, so he was looking for it.
Yeah.
But it was also the opportunity for us to go, oh, well, wait a minute, actually, is there something special about those E. coli? So we were lucky enough at that time, or I shouldn't say lucky, I would say good enough. We were good enough to be able to get some funding on that from Healthcare Research Wales. And so we collected lots of E. coli bacteria. And we did a few things with them. Firstly, we put them through our beautiful whole blood model, which is essentially taking blood from healthy volunteers and looking at the really early responses, because that's what's really important, is the early responses. So we did a little bit of that. We also had a little bit of patient data, so their mortality, their morbidity, male, female, and so on. And we also looked at the bugs themselves. And we looked at maybe some of the genes that the bugs had, and also how they grew in different media. Now, when you've got those 3 bits of data, so you've got the whole blood bit, you've got the patient data, and you've got the bug data, you can start to make associations.
Yeah.
And so when you start to make associations, you can start to go, oh, wait a minute, we've got a gene here in the E. coli bacteria that we know, oh, wait a minute, is associated with morbidity, or maybe is associated with the genes in the whole blood model. So we can start to make little associations and maybe then start to generate our own hit list, if you like, a hit list of narrowing down what are hundreds and hundreds, even thousands of factors right at the start of the immune response. And so what we start to then go on to ask the question is the links between some of those factors and what's happening in the clinic later on, and what's happening to multi-organ failure, and so on.
So is that how you went on then to study how you identified the...
Pretty much, yeah. So we got that. It was part of a PhD. So that PhD was involved in identifying those associations from those 3 different sets of data. And so what we had is a few lists of bacterial genes, but also human genes that we think maybe are more associated with that invasive capability of going from the local infection to the blood infection. And so what we have now is a set of, or short list, if you like, a short list of genes that we want to test, in the clinic. So our next stages really would be to really do that in patient samples. So we've done it in the setup in the lab, but now, wait a minute, is this true? Can we really detect these in patient urine, in patient blood, during an infection when people present in the intensive care unit?
So is it also identified whether certain people are more likely to get it or yeah?
I mean, this is one of the things that we'd really like to do. And I think this is maybe what's different. A lot of people, I think, have worked on multi-organ failure, and that's bad, obviously.
Yeah.
But actually, once that happened, you're then supporting the patient. What we want to do, and I think what a lot of people are starting to do now, is to look at pre-sepsis. So a state that is so early that maybe you've just got a few inflammatory markers and there are a few changes, and maybe even a few subtle changes, that ultimately you can say, well, wait a minute, if marker A is up, you can go on and say, well, wait a minute, those people do particularly badly when they're treated with this particular antibiotic. Or if this one's down, and so on. You can start to make little groups of patients that go on and either survive or die, but then you can change the treatment. So that's what we're really looking at, is this pre-sepsis state, and really trying to understand the changes in the immune responses, and also the changes in the bacteria early on. Because what the bacteria is doing maybe in that local body compartment to get to the blood might be very different to what it does once it's in the blood.
And that can vary.
And that can vary. And so maybe trying to assay and measure some of those things. So we got some targets.
Yeah.
Now it's really using those targets either alone or maybe in combination, because they might give us a stronger association with maybe particular outcomes. So that's pretty much where we are and we're hoping to go.
So is it this pre-stuff that kind of makes your findings so significant?
I think so. And I think probably when we started off, there was less research on the pre-sepsis. Now there are things like endotypes, where people have gone, well, wait a minute, we know if people come in and their temperature range is X, or their temperature range is Y, we know that those people can go on and have a particular outcome. We know now that, oh, well, wait a minute, if we've got patients that maybe respond to fluid resuscitation in some ways, some do better than others. But those sort of phenotypes right at the start are ways that we can start to, are ways that other researchers have tried to rank and group patients that come in on sepsis. But the problem is there's a big heterogeneity. There's a huge variation in the types of patients that come in and present. So, grouping them into guys that we think, oh, well, wait a minute, they've got this phenotype, they will go on and do very well if we do X, then that's what we're really trying to achieve in this pre-sepsis angle. And I think we're starting to really try to characterize that pre-sepsis state, which I think has always been there, but I think there's been less focus on it and more on maybe the multi-organ failure.
It's almost becoming like you're not reacting to it anymore. You're being proactive in how you can sort of...
Yeah, and you've got a prophylactic chance of intervening early.
Yeah.
Yeah.
So this pre-sepsis stage, is there potentially changes that could happen within the NHS for little or no extra cost that,
So I think so. I mean, that's the discussions to be had. But look, for us, some of the changes, and I think we've seen this a little bit in COVID. Some of the interventions you can do are quite simple, really.
Yeah.
So for instance, you've seen the little lateral flow tests.
Yeah.
I mean, those are just ELISA, so a technique we use to measure proteins in the lab. And what they've got on there is an ELISA against bits of COVID, essentially. But you could also have a lateral flow test for the different markers that we've identified, or a few of those different markers that others have identified. So you make a little panel. And of course, lateral flow tests are used all the time, everywhere.
Not invasive, is it?
And it's not invasive. But also there's things like PCR. And PCR's been used in the NHS, but in every laboratory, everywhere. And it doesn't matter how big the university is, doesn't matter how small the university is, doesn't matter how big the hospital is or how small the hospital is. They're all used these sort of techniques. And PCR is just a very simple way of amplifying up a very specific bit of nucleic acid. So, of course, you would normally use it for viruses and things like that. However, you can also use it, depending on the sensitivity, and the sensitivity is something to work on. You could use that for key bacterial genes that we've been looking at.
It's quite a practical pathway, isn't it, from your findings into what could potentially be real-world diagnostic? Yeah,
pretty much, I think, and keeping it simple, keeping it, I say cheap. You always want investment. So, if any of your listeners want to invest, please come and see me. Obviously.
Yeah, please reach out.
Please reach out. Nothing, obviously, it can be expensive. But, I think some of these solutions are the next step. And I think investing in these isn't a big investment in money. There's a little bit of investment there, but I think they're likely to work as well.
And like you said, it's that pre-stage, which makes a huge difference because you're not trying to...
Yeah. You're trying to really categorize those patients that will take a particular route. And it might be that we only go to one group of patients that is maybe a minority of all this. But it also could be a big group of patients. And because it's so early, I think it's a great intervention for the future.
So talking about the different groups of people, obviously with being like the Healthy Aging Research Institute, is there can you explain a little bit about how that early detection will help the older population?
Yeah, so one of the things we don't talk about in sepsis is post-sepsis scenarios. So we always talk about them. We always talk about the people with sepsis dying relatively quickly, and oh God, there was a surprise. But we always talk about the deaths, but we don't really talk about the survival.
Yeah.
And probably the best example of the survival that is related to post-sepsis syndrome are maybe some of the news articles you've read on long COVID. Because essentially COVID is, if you like, is a form of sepsis. So in other words, you survive sepsis or you survive COVID. And your life is particularly difficult because, you've got mental issues, anxieties, depressions, blurred vision, lack of confidence, frustrations, and you have to live with this for the rest of your life. So think of that now as your survivors, but if we could predict really early the ones that would survive, then actually you can start putting support in place. Well, wait a minute, this guy's very likely to survive, so actually he can go on the pathway for follow-up and looking after and dealing with maybe anxiety and maybe some of the cognitive issues that you get in the future. So really, from the terms of aging, I think this is the sort of thing that really we could be looking at. Because remember, most people that have sepsis, that it's not a linear curve from babies and upwards. It's actually a U-shaped curve. So a lot of babies get sepsis when there are issues, but also extremes of age. So in other words, older people. So in our cohort, we've got one or two that are 40, 50. Otherwise, they're all 70. And I think we've got somebody at 103 in there.
Oh my gosh.
So actually, it's the extremes of age. So once you've survived that, helping these people cope with the confusion that is post-sepsis and long COVID. And of course, we already have a bit of experience of dealing with long COVID, and it's similar things in the post-sepsis syndrome.
Because you do forget as well, don't you? Unless you're that person living in that moment and living with it, it's easy to just not really understand. I guess it's an awareness thing, isn't it? What does life after sepsis look like for some of these people? Are they always going to be prone to?
And they are. They're going to be prone to reinfection and readmission to hospital.
And that in itself is quite, and that as well is a cost as well, isn't it? If you want to talk about cost, that readmission. So with that in mind then, in the real world context, what difference could this make?
Yeah, so I think if we were going to be practical, it's the triaging when people come into a hospital. That is where I think we could ultimately help. And that's why maybe the next step is looking at longitudinal studies in patients. So those triage questions are really important because time is important in sepsis. So there's a correlation between the time to antibiotic treatment or time to give antibiotics and death, pretty much.
Yeah.
Okay. So if you can change that time and improve that time and start to group patients for treatment and therapy earlier, can make a huge difference, I think, in the clinic. So basically, that triage assessment when they're doing that and putting people into groups, and that might be some of the groups that we're thinking of, but also some of the groups that are already, out there in the literature, like I said, with temperature and fluid resuscitation. If we can start doing all of those and start pooling together data, we can start to create newer groups, more practical groups, and groups that are more accurate at predicting where patient outcomes.
So you kind of touched on it. Is that what your next steps are?
So next steps have to be, my wife and children always say, "Stop faffing, Tom." So I got to write the paper. And it is written. It's 80% done, but I've got to get it over the line. So there's that. There are a few little interesting more on the microbiology side than maybe on the biomarker side. So that's number one thing. Number two things is that we've got the next phase in funding. We have to do that, and it would be more of a longitudinal, or more of a cohort study in patients. But the other thing that I've sort of ignored and probably didn't take seriously over the last 20, 25 years, and I apologize to these guys, is the IP and the commercialization. And I've talked to lots of guys in the university on this, and I probably didn't take it seriously. So probably because I didn't think maybe there were possibilities with it. But now, having talked to people and probably understood it a little bit more on the license and spin outs, I think some of this would benefit from that.
Yeah.
So we're also looking at potential licensing and potential turning either the biomarkers or combinations of biomarkers into products. So that's pretty much where we're going. Again, it's early days, but I think we're hopeful.
Yeah.
We're hopeful. So, and like I say, if any of your listeners are there listening and they're interested in any part of this, whether it be from a patient, perspective, because we're interested in talking to patients, in particular those long COVID
Long,
patients,
yeah.
but also maybe in some of the technologies that, might put a molecule or a nucleic acid into a product or into a PCR test or into a lateral flow test, please contact me or the group. We're a happy bunch. We we have our lab meetings. Please come along and out of that. And, we can learn from each other.
And that's the thing with research, isn't it? I've said this, I think, so many times, but every time I sit here, I'm just fascinated. I'm like, wow, every time, because it's just, the possibilities are literally endless, aren't they?
Yeah.
And the difference that it could make is just huge.
Completely. And you've got to remember, I think, probably gone are the days where you can sort of beaver away in the lab and work on this one molecule all your life. And now, it's about societal impact.
Yeah.
And I think, why wouldn't you want to do that?
Yeah.
Why wouldn't you want all your research to be turned into something that's helpful to society? You wouldn't. I think everybody wants that in the end. How to do it, is more difficult, but we've got people to help us in the university environment. So.
And there's a lot of that interdisciplinary stuff happening, isn't it? You've got, like you mentioned, you've worked with someone from
the whole of our health completely.
And it's that in being able to spread it wider. And I guess it comes down to that awareness side of things as well, doesn't it? Oh, I've got so many more things we could speak about, but unfortunately, as interesting as this topic of conversation is, when we get to the next stages, you're going to have to come back.
I will, obviously, yes.
But also, if people do want to reach out, they can contact you via your staff
email, phone call, Zoom, anything you like, or even just pop out at the 5th floor in ILS 1. I don't know how far you'll get. You might get out of the lift. You won't get into the lab, obviously. But, yes, obviously.
So maybe check in via email first.
Yes.
And you can reach out to us at the Healthy Aging and Chronic Conditions Research Institute if you want to. But I've really enjoyed it. That's just fascinating.
I have as well, Adrian. It's been fun.
Thank you so much for coming in and talking to us.
Okay, thank you.